Hello, everyone. Welcome to this screencast organized by the Royal Society of Chemistry Early Career Network of Manchester and District section. First of all, thank you very much for the invitation. And today we're going to talk about biochemical process design. Specifically, we will discuss different digital techniques that can be used to quantify biochemistry knowledge and then apply it to the real world. Now, first of all, I would like to briefly introduce myself. My name is Dongda Zhang. I'm a chemical engineer. I got my bachelor degree at Tianjin University in 2011, and then I moved to Imperial College for my master's study. And in 2016, I got my PhD degree at the University of Cambridge. Currently, I'm a university lecturer at the University of Manchester, and also I'm an honorary research fellow at Imperial College, London. My research mainly focuses on process systems engineering and machine learning. And in my group, we mainly develop different industry-focused mathematical modeling techniques for chemical and biochemical process digitalization. So many modeling tools in our group are focusing on biochemical and chemical process prediction modeling, optimization, design, and control, as well as scale up. Now, when we talk about chemistry and consider its real-world application, I believe many of you will come across with the idea of chemical engineering. And when you're talking about chemical engineering, I suppose many of you will think of petrochemical industry. As a trained chemical engineer, this image is my understanding to petrochemical industry, which is a very well organized and very well designed, very modern chemical plant. But also at the same time, I understand many of you will consider petrochemical industry as environmentally destructive and even dangerous sector, which is true, because for a petrochemical industry, many of its raw material comes from petrochemicals, which are non-renewable resources. So it's not really sustainable. And also a chemical plant will have a lot of pollutions and emissions. So it also causes some environmental impact. And because a petrochemical reaction usually happens at high temperature, high pressure condition, so there are also some risks in the safety of the process, and we often hear some incidents happening in the plant, like explosion, right? So now, moving to the future, we would like our process to be more sustainable, more environmentally friendly, and also safer. As a result, within the last 10 years, there are more demand about using biochemical processes to replace the traditional petrochemical processes, mainly because biochemical processes are operated under room temperature, room conditions, so it is safer, and we have more resources like biomass waste, which could be sustainable and could be renewable. And also, we believe that the environmental impact of biochemical process could be less than petrochemical industry. So now, what is a biochemical process and what biochemical process can produce? In fact, biochemical process has been also widely used in many different countries, and we can use it to produce so many different products. For example, we can use biochemical process to produce biofuels. And the advantage of biofuels is that we basically first, we can use solar energy and carbon dioxide to produce sugar through photosynthesis. Okay, and then we can use this sugar to produce biofuels. So when we burn biofuel, we actually produce carbon dioxide, which will be fixed by photosynthesis again, and then it will be used to produce biofuel again. So this biofuel is actually carbon neutral. It doesn't produce any additional carbon dioxide. Also, we can use biochemical process to produce food and drinks, like beer. It comes from fermentation, and that is a common sense. Many of us, we know that, right? And finally, we can use biochemical processes to produce some important pharmaceutical and nutraceuticals, which are related to our health. And fundamentally speaking, independent of the product we want to produce, all of the biochemical processes are related to microorganisms, okay? And we are actually dealing with a metabolic reaction at work. So from the biochemistry perspective, it's quite important for us to understand the kinetics of these metabolic reactions involved in the metabolic reaction at work, because this is the actually highly regulated reaction at work that convert our material like glucose, sugar, and carbon dioxide into our desired product like biofuel, food and drinks, and some nutraceuticals. However, again, this metabolic reaction work is only the biochemistry knowledge and focusing on the intracellular level. But when we're applying this biochemistry knowledge into the real world, like we want to industrialize biochemical processes, then we will be looking at the industrial operation. And a big question that we have to ask ourselves is that, are biochemical processes really ready to replace petrochemical industry? Now, let's look at some facts. First of all, compared to a petrochemical process where the reaction conversion can be over 90%, the raw material to final product conversion in a biochemical process is often lower than 20%. And that is because of these complex metabolic reaction networks. So when microorganisms receive raw material like glucose or some other kind of sugar, it will convert glucose into our desired product through this metabolic network, but also at the same time produce lots of byproducts which are not desirable. As a result, only 20% of the sugars can be converted to useful product. The remaining 80% are wasted. Now, so we know that if we really want to make biochemical process more competitive and more efficient, we have to understand the biochemistry knowledge even better and then design new mutants. The second challenge is that when we look at scale up of this biochemical process, rather than implementing some experiments at the lab scale, but actually try to implement the experiment or the process at industrial system, then we have to design this large scale bioreactor, which can be very challenging. Because when we are scaling up the process, you will find that other factors such as fluid dynamics and mass transfer will start to influence the whole system. So we know that, for example, microorganisms like yeast and bacteria, their growth is very sensitive to the cultural condition like pH, temperature, and nutrient concentration. In a large-scale bioreactor, if the culture is not very well mixed, then in some of the part of the reactor, we can have like very high pH or very low pH, which is really preventing cells growth, and that can cause even the entire batch to fail. So these are some big issues which we have to deal with. Just how to develop high efficiency bioreactor is a big challenge. And we also need to understand under this big bioreactor system, what is the most efficient mutant that we can design in order to maximize the conversion of raw material, right? So this is very important challenge that we have to solve. However, even if we solve all the technical issues, it doesn't mean we can apply biochemical process to the real world. Why? Because there's another factor that we have to consider, and that is economic competitiveness. An example is ABE fermentation process, which is to use fermentation to simultaneously produce acetone, butanol, and ethanol. We know that butanol and ethanol, they are biofuels. This process actually has been developed for over 100 years old. For over 100 years, it was first proposed in 1860s. Until now, the technical challenge of this process has been almost completely solved, but the process is still not commercially viable. Why? The reason is because if you look at the market, then the price of butanol and ethanol from petrochemical industry is much cheaper than that from ABE fermentation system. As a result, for biochemical process, they are currently still mainly used to produce high-value bioproduct, and they are not ready to produce biofuels. If we really want to design competitive biochemical processes which can be used in the near future, then we have to first understand the biochemistry knowledge, including both intracellular metabolic reaction kinetics, where we only focus on one or two, like very few cells, right? And at the macro scale, under the macro scale, biomass growth and product synthesis kinetics, which is the interaction between biomass growth and the cultural environment. Like in a lab scale reactor, we may have 10 to the 4 to 10 to the 6 cells. We also need to understand the biochemical engineering knowledge, which focusing on the scale up and the design of the bioreactor. And in industrial fermentation process, we may often have 10 to the 9 cells, and we know each cell has thousands of metabolic reactions. So fundamentally speaking, we are dealing with a huge number of reactions, and it's very complex reaction network. We also need to consider the economic aspect of the process. So it means that we need to consider how to design the separation process to make sure it is not very expensive, and then like the entire process investment can be as cheap as petrochemical industry. So to solve all of these challenges, one thing we can do is to design this kind of in silico biochemical process by using the knowledge from process system engineering and machine learning. The main concept is that we can first quantify and digitalize each individual component from different aspects, including both biochemistry and knowledge. And then we can combine different models at a different level, from the intracellular metabolic level to the macro scale process level until the entire plant level. Then we integrate these models to build a multi-scale model, and then use this multi-scale model to accelerate bioprocess development. This is basically the concept of the fourth industrial revolution, including process automation and process digitalization. So it definitely sounds to be a very ambitious and a very complex procedure. Now, we know the high-level idea, and we know why it is interesting, why it is important, and why it is novel. But another question we want to ask is that, how can we actually digitalize and quantify this biochemistry knowledge and biochemical engineering knowledge? Well, in fact, there are different techniques we can use. I will give you some simple examples. For example, in one of our research, we want to study and understand how to use microalgae for biofuel production. So basically, microalgae can conduct photosynthesis and then convert carbon dioxide into biodiesel. Now, if we want to simulate and quantify this process, there are different approaches that can work. The first one is to use biochemical knowledge. If you ever learned chemistry and reaction engineering, then you know that many times you have different reaction kinetics derived based on physical knowledge, right? In biochemistry, we have the same thing. So we also have a kinetic model which can be used to simulate biomass growth and product synthesis. And this model usually looks very complex. It includes that intensity, carbon salts, nitrogen salts, and some other influential factors. And the specific expression is always derived based on biological knowledge or based on experience. However, compared to a chemical process, biochemical reaction is much more complex. As we mentioned, it always involves thousands of metabolic reactions. So a pure biochemistry kinetic model is always of low accuracy. Now, if you look at the simulation result of this kinetic model and then compare it against experimental data, you can see that there are always some high model plant or model process mismatch, right? So this model is not accurate enough to actually quantify the original process, and we can't really use it for decision making, and we can't really use it to accelerate the design of biochemical processes. So how can we improve the model accuracy? Well, there are different approaches. The first one is to build a data-driven model using the knowledge from machine learning or artificial intelligence. Imagine we can actually build a data-driven model to learn from existing bioprocess data, so we can feed the biomass concentration and culture condition at the current time step, and then use this data-driven model to predict biomass concentration at the next time step. But usually this data-driven model or machine learning model can give us very high accuracy in terms of simulation. So for example, in this figure, you can see that this red point are experimental data, and this blue point data-driven model simulation result. You can see that this model simulation is much more accurate than a biochemical kinetic model. And there are different data-driven models we can use, including artificial neural network, enzyme learning, and Gaussian processes, which I won't go into detail. However, as a biochemist or chemical engineer, it's not the data that we're looking for. We are mainly interested in the knowledge behind the data. So if the model we construct cannot provide us some extra biological knowledge, or cannot really be used to generate new knowledge, then it's not really useful to us. As a result, in order to generate biochemical knowledge and at the same time get a highly accurate model, we can actually use hybrid modeling to basically combine physical knowledge with machine learning technique. Like one example is that we can use kinetic model to simulate the bioprocess, and then we use a data-driven model to rectify the model process mismatch. In this case, you can see that the model is able to actually give us very accurate prediction. And you can see, for example, in this case, the lines are model prediction and the points are experimental data. You can then use this high accurate model to predict different scenarios, including different temperature, different nutrient supply strategy to maximize the process conversion. So this is a simple example how we can actually use biochemical kinetic model or data-driven modeling techniques to quantify a bioprocess. We also need to know biochemical knowledge, not just biochemistry knowledge, right? We need to understand the engineering challenge, which is the scale-up of the process. So is there any technique that we can use? Well, the answer is yes. In one of our experiments, in one of our projects, we actually want to design a large-scale bioreactor and at the same time optimize the reaction condition, like the bioreaction condition for biofuel production. So this project actually is collaborated with Imperial College London. We basically want to optimize that intensity, temperature, and the gas inflow rate, which provide carbon dioxide growth. And also want to optimize want to design a pilot scale bioreactor, in particular, the spudger of the reactor. So at the same time, we need to consider some practical process constraints, as we mentioned, the fluid dynamics. In this case, it's the friction velocity, because if we have a very high friction velocity in the culture, then the culture will tear up cells. So it will cause the decay and death of cells. In this case, now, by using the traditional chemical engineering knowledge, we can build a kinetic model to simulate the bioreactions. And then we can build a CFD model, which is computational fluid dynamics. That one can be used to simulate the fluid dynamics of a large-scale bioreactor. In principle, we can combine these two models to build a multi-scale model and use this model to predict and to optimize the reaction condition and the bioreactor condition. But in reality, this is infeasible because to run this model, it will take 1 week time just to run for each run. And if we really want to do optimization, we have to run thousands of different scenarios, which if you calculate the time, it may take months, 2 years. And that is just impossible, right? How do we solve this problem? Well, we can build a surrogate model to reduce the computational time cost. The concept of surrogate model is that we can actually use the data generated from the rigorous model, like in this case, the image generated from the CFD model, to train a machine learning model, such as convolutional neural network, which is image reading technique. And then we can directly optimize this neural network. And it is very efficient to construct this surrogate model, and the optimization will only take 2 hours. Once we get an optimal solution, we can then compare it with the rigorous model's verification result. And in many cases, I can tell you this digital model is highly accurate. So by using this kind of surrogate modeling technique, we're able to design efficient large-scale bioreactor to facilitate the scale-up of a bioprocess. And we're able to reduce the time cost from months or years down to days or hours. Now, if you look at the future of biochemistry and particular biochemical engineering, which aims to apply the knowledge of biochemistry into the real world, I can say that at this moment, for each individual level, from metabolic level to the process level and to the plant level, there are already advanced digital techniques that has been developed, and we can build different models for each individual sector. But if we really want to implement this whole system integrated approach, then we must link all of the knowledge between different scales. And then this is a multidisciplinary research combining knowledge from chemistry, engineering, biology, and data science. So as a suggestion for early career researchers, definitely I would say conducting multidisciplinary research and establish collaborations is the most important thing to enable the design of novel biochemical processes and allow its early implementation to the real. Finally, I'd like to say that many of my research are conducted with collaborations at Imperial College, UCL, and Xiamen University in China. So thank you very much for your listening. If you are interested in my talk, you're always welcome to contact me. Here's my email address. Thank you.